Hey guys, welcome to AP Biology, and we're going to be focusing in on the Calvin cycle, i.e., the dark reactions, i.e., the light independent reactions. So you might see those three different names up there, but most proper name is probably the Calvin cycle, but you never know what you're going to see. All right, so yes, the College Board is not as forgiving as I am, so this is something that you must watch and take your notes out. Make sure you have your notes out for this particular section. We're actually going to do two lectures and put them together real quick here today. I'm going to try and keep it slow, quick. All right, so first of all, we are going to be focusing on the second or the right-hand side of this overview, the Calvin cycle. And let's just look at it very briefly. We are going to use the two products of the light-dependent reactions, ATP and NADPH, and we're going to take some CO2 from the atmosphere, and we're going to use the energy of ATP to rearrange the molecules, and we're going to use the electrons being carried by NADPH to reduce carbon dioxide. So remember, reduction is gain. We're going to take the electrons away from NADPH. That means we're going to oxidize NADPH and use those electrons and attach them to carbon dioxide so we can make sugar. So remember, every reaction is a combination of oxidation and reduction. Redox reactions is another name that you might hear for those. All right, so you don't need to know all the details here. I will tell you what you do need to know. So first of all, Calvin cycle can be divided into three steps. Phase one is carbon fixation. What does that mean? That means that we're taking inorganic CO2, and again, it's not organic because it does not have hydrogen, and we are going to add electrons to it, and we're going to also basically incorporate it into an organic molecule. We haven't added the electrons yet, but we are going to incorporate it into an organic molecule and use some energy in order to do that anabolic process. Remember, this is an anabolic process. So you need to know the input, CO2. Where's the CO2 coming from? It's coming from the atmosphere. How does it get into the plant? It comes in through the stomata, or the little holes on the bottom of the leaves of the plants. Okay? Different plants, such as cacti and things like that, have similar structures, although not in the same place, because cactuses don't really have leaves, although they really do. The leaves are just been modified into spines. Okay, but anyways, so a couple of things you need to know. You need to know the name of the enzyme that catalyzes this reaction, and this is one of those enzymes that does not end in ASE. Rubisco. Sounds like Nabisco, that makes cookies. Okay, it's rubisco. It is probably the most abundant protein on the planet. And it's going to basically take a 5-carbon molecule called rubip, that's actually ribulose bisphosphate. So ribulose, that means it's a sugar. We're going to take this 5-carbon sugar and we're going to add a CO2 to it. Okay, and we're going to make what's going to happen is it's going to make a 6-carbon molecule, but that is very quickly broken down into 2 3-carbon molecules called phosphoglycerate, or PG. I think it's PGL, PGAL. So it's what it's called. So you need to know the name of rubisco, the enzyme. You need to know the name of the input, which is CO2. And I always tell my students you should probably know the 5-carbon sugar name of rubip is how we pronounce it. Okay, so phase one, carbon fixation, turning it into an organic molecule. Stage two, or phase two, is going to be reduction. Here is where we are going to use the electrons of NADPH to reduce phosphoglycerate into glyceraldehyde 3-phosphate, okay, G3P, which is a sugar, even though it doesn't end in OSE. Okay, I know that, but it is a sugar. Okay, so we're producing a sugar in that output. I do also want you to know that we're starting off with 3 molecules of rubip, 3 molecules of CO2, and you'll see 6 molecules of phosphoglycerate, 6 molecules of 1,5-phosphoglycerate, and 6 molecules of glyceraldehyde. If you counted up the carbons, you'd have 15 here, plus 3 equals 18. 6 times 3 is 18. 6 times 3 is 18. 6 times 3 is 18. One comes out, so we're now back down to 15 carbons. One is going to be a 3-carbon sugar, and that 3-carbon sugar will be combined with another 3-carbon sugar to make glucose in a separate metabolic pathway. All right, so phase 2 is reduction. Also notice the number of ATPs we have currently used. We've used 6 ATPs and 6 NADPHs, so now they're equal, they're balanced. And the final third stage is going to be regeneration of RUBEP. Okay, that's phase 3, the regeneration of the CO2 acceptor RUBEP. And what we're seeing is that we're taking, remember, we had a total of 18 carbon molecules at this point. We lost 3, so we're down to 15. We now have 5 of these G3Ps, okay? And now we're going to add some more ATPs. Okay, and that's going to turn the G3P into ribulose bisphosphate, biphosphate. There's the 2 phosphates, one there and one there. Okay, so now notice we've now used a total of 9 ATPs and 6 NADPHs, and this is the reason why plants also do a combination of non-cyclic and cyclic electron flow. So if you remember, with non-cyclic electron flow, you make both ATP and NADPH. But with cyclic electron flow, you only make ATP, mainly because the process of the Calvin cycle uses more ATP than it does NADPH, so we have to make it someplace. And that is basically the Calvin cycle. All right, it's not too bad, but I do want you to notice the essential components are boxed. So you need to know the CO2, the ribisco, the enzyme, where the ATP and NADPH are being used. You should know the 3 phases, phase 1, phase 2, and phase 3, and then the output. Okay, that's what you need. You don't need to know all these little names, although ribulose bisphosphate you should know, okay, or RUBEP. And that's giving you some sugar. All right, so again, there's photosynthesis, the big picture. These are some links that you can look on. I will put these on in Schoology, so you can click on those links and check it out. All right, so I do want to look at a little bit of the structure of leaves. Okay, so this is a typical structure of what we call a C3 plant. Most of the plants you look outside are going to be a C3 photosynthesis. Why is it called C3 photosynthesis? Because the first sugar that's being made is G3P, a 3-carbon sugar. Okay. But anyways, this is a cross-section of a leaf, and the top of the leaf has a waxy cuticle layer that helps to prevent water loss and protects the plants. Then we have this palisade mesophyll. This is where most of the chloroplasts are going to be stored. And then we have this area called the spongy mesophyll, and this is where gases and water and things like that are oftentimes stored. And then in the bottom of the leaf, the lower epidermis, you do have these stomata. What is a stomata? It's a hole in the leaf that has these specialized guard cells that can open and close on the base of the leaf. They are open when the plant has enough water and it needs to get CO2 in. And when the plant becomes water stressed, those stomata close. And that's going to lead to some problems. Okay, so do you notice what's going in the stomata? CO2 is going in the stomata. What's coming out? Water and oxygen. Okay, water and oxygen are coming out. CO2 is going in. Okay, so let's think about this for a second. All right, there are other types of photosynthesis. What we talked about was something called C3 photosynthesis. There is another type of photosynthesis that occurs called C4 photosynthesis. C4 photosynthesis evolved later, and it's a process that basically tries and circumvents a problem called photorespiration. Okay, so think about that. That's light and then respiration, as you would see in a mitochondria, where the organic molecules are being broken down to provide energy. So let's think about this for a second. In photorespiration, when a plant becomes stressed for water, those stomata on the bottom of the leaf are going to close. So if those stomata close, what can no longer get in? CO2. CO2 not get into the leaf. Oxygen cannot escape the leaf. Water can't escape the leaf. So if that plant is water stressed, it wants to close those guard cells so water doesn't leave the plant. But CO2 does not get into the plant and oxygen builds up. So why is that a problem? Okay, let's go back to here. This enzyme ribisco. Photosynthesis most likely evolved, we think, somewhere between 3.6 and 3.8 billion years ago. And this is brand new data that just came out this year, actually, 2021. And Rubisco evolved at a time, photosynthesis evolved at a time where there was no free oxygen in the atmosphere or much free oxygen in the water where life was evolving. So that causes a problem because Rubisco, the active site of Rubisco, has an equal affinity for both CO2 and water. Okay? Excuse me, not water, but oxygen. So it has an equal affinity for both CO2 and oxygen. So if there's a lot of oxygen in the environment where this process is occurring, the Calvin cycle, then oxygen has an equal chance of getting into the active site of Rubisco. Well, if that happens, then we're not going to make this 6-carbon intermediate molecule. Instead, we're only going to make a 5-carbon molecule. Instead of having 6 3-phosphoglycerates, we're going to have 1 3-carbon and a 2-carbon molecule. Well, that 3-carbon and those 2-carbon molecules are going to be both broken down to provide energy instead of making sugar. So photorespiration, which is a result of the stomata being closed, and when the stomata are closed, then the CO2 levels are going to go down because the rubisco is going to use up most of that CO2. The oxygen levels are going to increase inside that leaf because water is still being broken down through the process of photolysis. And now, instead of undergoing the process of making sugar in the Calvin cycle, we're going to be using existing carbon molecules of the Calvin cycle and breaking them apart in order to just going to be broken down. So mass will not be accumulated, instead it will disappear. So photorespiration is a huge problem in modern agriculture, especially as our global temperatures increase and we're getting all these wacky, intense rainfalls in some areas and extreme droughts in other areas. It's causing lots of problems. Well, this has happened in the Earth's past before, and another means had evolved called C4 photosynthesis. And what happens with C4 photosynthesis is that the process actually takes place in a couple of different places. The primary fixation of carbon takes place in the cells that we call the mesophyll cells, and they also have a different architecture than a C3 leaf. Notice that the mesophyll is arranged in this bundle. And instead of having rubisco, there's an enzyme called PEP carboxylase, which stands for phosphoenol pyruvate carboxylase, but you don't need to know that. But PEP carboxylase has a great affinity for CO2 and no affinity for oxygen. So even if oxygen levels are high and CO2 levels are low, PEP carboxylase will still fix carbon into a 4-carbon molecule called oxaloacetate. And that's the reason why it's called C4 photosynthesis, because the first molecule being made after carbon fixation is a 4-carbon molecule. Okay, and this is happening in a different cell, and then that 4-carbon molecule is going to get delivered to a specialized cell called the bundle sheath cell, where the carbon is given off as CO2, and then normal Calvin cycle stuff happens. So instead, we have we have carbon fixation occurring in a different location than the Calvin cycle, whereas in normal C3 leaves, it all happens in the same place. And this just enables a plant to live in an environment that is a lot hotter because it's going to preferentially take in just CO2. Or I should say the enzyme's only going to fix CO2. There's also another type of plant called a cam plant, which is like pineapples and most grasses. And what these guys do is they do something very similar to C4 photosynthesis, except they close their stomata during the day and they open them up at night. And at night, they fix the CO2 and they make organic acids. And during the daytime, when the stomata are closed, those organic acids are going to be used to release CO2 in the Calvin cycle. So those guys have a slightly different method. I just want you to be aware of those. You don't really need to know the details of those at all, but I do want you to know that evolution and natural selection have evolved different mechanisms for dealing with plants in different types of environments. Okay, so let's go ahead and look at the big picture of photosynthesis. Again, you've seen this diagram before. Let's just look at it again, break it down. I want you guys to know this. Remember, the main energy source for our planet is the sun. The plants, the primary producers, and the algae and the protists and things like that are able to capture that in the process of photosynthesis and provide energy for all of the other heterotrophic consumers in different chains. Remember, energy flows one way through the system, and every time the energy is transferred or transformed, some of that energy is going to be given off as heat energy, and eventually all that heat energy will leave our planet, okay? And the most important thing is, again, that the matter is being recycled. And notice that the energy moves from one group to another. These are trophic levels: the primary producer trophic level, primary consumer trophic level. These would be your herbivores. Secondary consumers are going to be your carnivores, omnivores, things of that nature. Tertiary consumers and above are generally going to be your top level or apex predators. Everything dies, forms into detritus, which is then broken down to recycle the nutrients. So any ecosystem must have producers and detritivores, things that are going to recycle the nutrients. Those are the two most important components of any ecosystem: the producers and the organisms that are going to recycle all those dead and dying materials in order to get those back into the system. Okay. Let's quickly look at the carbon cycle. Also, where does the carbon come from? The main storage area for carbon is in the atmosphere for short-term carbon usage. The process of photosynthesis is going to take that carbon out of the atmosphere and convert it into living material. And then the process of cellular respiration by both plants and animals and decomposition are going to return that CO2 back to the atmosphere. Now, long-term carbon can be stored in the ground for long periods of time in the form of fossil fuels such as coal and oil. Okay, but our massive burning of these fossil fuels has led to an overload of carbon in our atmosphere. Okay. Oh, just want to go back here once again. Remember, the reason why these boxes start big and get smaller, that's referring to the amount of energy available in any trophic level available to the next trophic level above it. And we've already talked about the 10% rule about this. Okay, so only about 10% of the energy of any trophic level is available to the next trophic level. And once again, this is the reason why your third and fourth level consumers, your apex predators, are always rare, because there's just not enough energy in the system to support all of them. So that's why those numbers of those organisms are always less. All right, so here's a 10% rule with a caterpillar. And if I was able to take a caterpillar and look at how much energy that particular animal had available, we see that there's 200 joules of energy in. A joule is just another term for like energy, like a calorie is. 200 joules in the plant material eaten by the caterpillar. 100 joules, or 50% of it, will be lost in feces. And if you ever kept a caterpillar, you can obviously see that. 67 joules will be lost as cellular respiration. Again, that's not leading to growth or anything of that nature. That's just basic cellular processes. And only 33 joules of that original 200 grams will be used for growth. That actually comes out closer to about a 15% transfer of energy. And again, that 10% rule is just a general number. It's going to vary from maybe a max of 20-25% down to like 1 or 2%. So 10% is our general number that we like to use for this. And again, looking at food webs, do notice up here that the the largest organisms, the baleen whales, are going to be eating some of the smallest organisms, where the toothed whales, which are eating seals and things of that nature, they don't get that big. Okay, your toothed whales just don't get as big as the baleen whales. Why? Because there's just not as much food out there, because these guys are eating lower on the food chain, which is a good argument for eating more plant-based foods as our population grows. Just more food available for us, for humans, than if we try to eat animals, because we eat animals, we got to feed them, we have to grow food for them, corn and wheat and things like that, and hay, and feed that to the animals, and then we only get a small percentage of that. So if we want to have more people on this planet, which we really don't, but if there are, to support them all, we should all eat a little bit lower on the food chain. Okay, this